Assalamualaikum friends, welcome back. Today I've got an exciting video for you, and that is on biofilm. But before starting the lecture, I like to tell you guys that these videos are meant for educational purposes. Things and treatments may change with time. If I get wrong or miss anything, your input is always welcome in the comments section. Alright, let's dive straight into the video. Biofilm. It has got two words in it. First one is bio, which means microorganisms or which refers to life. And the second word is film. Film is kind of liquid. What actually is a biofilm? It is a thin layer of microbial slime and proteins adhering to a surface. It is formed when microbes include bacteria, viruses, fungi, protists. When these microbes adhere to any surface can be a living surface or a non-living surface, we'll talk about that. And these microbes produce a matrix called EPS, the extracellular polymeric substances. And then biofilm is formed. What is EPS? EPS is a sticky type of glue-like material in which the microbes live. This is how a biofilm looks like. The outer green color is the EPS, and inside it there are certain microbes. These are bacteria, the cocci, spirals, and rods. Lecture outline. We are done with the introduction of biofilm and its diagrammatic representation. Now we'll be looking at the components of biofilm, the microbes responsible for forming biofilms, location of biofilms, causative factors, their developmental stages, and how the biofilm is formed, microbial benefits of biofilm, how we are going to treat them, and at the end, as usual, we'll review the lecture. Components of biofilms. A biofilm is made up of two components. Number one, microbes, and number two, the EPS, extracellular polymeric substances. EPS includes organic carbons, polysaccharides, these are the carbs, the carbohydrates, then proteins, lipids, nucleic acids, and other minor components. In microbes, we have bacteria, both gram-positive and gram-negative, protists, fungi, and viruses. These all are responsible for forming biofilm. In this picture, you can see EPS containing different types of bacteria, both gram-positive and gram-negative. And EPS is glue-like, sticky kind of material that holds these microbes together. Location. Biofilms occur on teeth as dental plaques, on skin, mucosal surfaces like of heart and lungs. They're also present on certain devices like catheters, for example, urinary catheter or venous catheter. They are also present in nasal packings, surgical lap pads, contact lenses used in eyes, prostheses, for example, prosthetic heart valves or prosthetic hip joints, and tampons. If these devices are left in the human body for a long time, then biofilms are formed. What are the causative factors that lead to the formation of biofilms? Microbes form biofilms due to the following factors: the cellular recognition of specific or non-specific attachment sites on a surface, nutritional cues, exposure to planktonic cells to sub-inhibitory concentrations of antibiotics. Planktonic means floating. And also the long stay of certain external devices in human body, as we've discussed earlier. Developmental stages. Following 5 stages are involved in the development of biofilm. Number 1 is initial attachment. Number 2 is permanent or irreversible attachment. Number 3 is maturation 1, or also referred as colonization. Number 4 is maturation 2, or we can also call it growth stage. And the final stage is development. Formation of biofilm. We are going to look how a biofilm is formed and developed. The first important thing that the microbe will identify the surface on which it is going to form the biofilm, whether it is a living surface or a non-living surface. In living surfaces, we've got mucosal surfaces like of heart and respiratory tract, and in non-living surfaces like catheters, prosthetic devices, tampons, surgical lap pads, etc. And the formation of biofilm starts with the attachment of free-floating microbes to the surface. Let's talk about the stages. The first stage, as we know, is initial attachment. It is when bacteria, after identification, attaches to the biofilm. not just bacteria, but any of the microbes. Here I'll be considering bacteria. This attachment is weak and reversible and is facilitated by van der Waals forces. As you can see there, the bacteria is being attached to this non-living surface through van der Waals forces. The second stage is permanent or irreversible attachment. When microbes or the microorganisms are not separated from the surface for a long time, they establish a more permanent attachment, as you can visualize there. Attachment is done using adhesion structures like pili or other appendages. This stage strengthens the connection between the microbe and the surface. The third stage is maturation 1, or colonization. As its name shows that it will increase in number, it will form colonies. So during colonization, the attached microorganisms communicate and interact. Signaling molecules like acetylhomoserine lactone AHL are used in process called quorum sensing. This QS process enables microbes to coordinate their activities, and also it is involved in the production of EPS. As you can see there, this is the colony of bacteria. Surrounding them is the EPS. This is produced due to quorum sensing process. Communication facilitates the formation of cooperative structures within the biofilm. The fourth stage is maturation 2, or growth. Biofilm grows in size and complexity after colonization. Growth is achieved through cell division and recruitment of more microorganisms. The biofilm develops distinct layers and channels. These structures aid in the flow of nutrients and waste products. The final stage is development. At this stage, biofilm is fully established, stable in shape and size. Structure of biofilm remains relatively unchanged in the development stage. Microbes exhibit specialized function and behaviors. They communicate with each other, they transfer genetic material, they divide there, they interact with each other. These contribute to collective properties like antibiotic resistance and survival in harsh environments. This leads us to microbial benefits of biofilm. There are many benefits, but the most important one is the protection. Biofilm protects the microbes from immunity, the harsh environments, from antibiotics. This is termed as microbial resistance, which means that bacteria are resistant to anything that can damage it. Not just bacteria, any of the microbes. Biofilm provides an hostile environment where microbes can communicate, live, feed, and grow. For example, it's 200 times harder to kill a bacteria inside a biofilm as compared to bacteria which is alone. Treatment. It's really hard to treat or eradicate the biofilm, but it is only possible when a biofilm is ruptured. And when we think biofilm will be ruptured, it will be ruptured when there are many microbes, let's say bacteria, present in the EPS, in the biofilm, and this layer ruptures to release all the floating bacteria to infect other surfaces. For example, if the bacteria wants to spread the infection through other parts of the human body. Removal of devices. For example, biofilm is present on the catheter. For example, biofilm is present on the urinary catheter. After removing the urinary catheter, the cause is removed, so problem will be treated. Proper dental hygiene. As biofilms are formed on teeth as dental plaque, in order to let them not form on the teeth, proper brushing, flossing, and using clean water. But if this happened for any reason, then restorations, non-surgical or surgical dental therapies can be done. Root canal treatments and dental implants can also prove very important in order to eradicate biofilms or remove biofilms from teeth. Antibiotics play a really cool role there. They are going to kill the bacteria inside the EPS. Certain antibiotics have that capability. Research has shown that probiotics also play a very important role in removing biofilms, and there are also certain foods that can help remove biofilms. Alright guys, let's review everything really quick. Biofilm, it is an aggregate of microbes in which cells, we are talking about the microbial cells, adhere to each other on a surface. They have got following developmental stages. Number 1, initial attachment. Number 2, irreversible or permanent attachment. Number 3, maturation 1 or colonization. Number 4, maturation 2 or growth stage. Number 5 is development. Biofilms develop resistance against antibiotics, immunity, harsh environments. Biofilm is composed of microbes and EPS. Location: biofilm are located on teeth as dental plaque, catheters and nasal packing, surgical lap pads, contact lenses, prostheses, for example prosthetic hip joints or prosthetic heart valves, skin, mucosal surfaces, and tampons. Treatment: the rupture of biofilm play an important role in its treatment. Removal of devices like catheters, proper dental hygiene, antibiotics. And that's it for today's video. I hope you guys enjoyed it. You've learned something. If you really did, comment down below in the comment section. And also, if you want to connect with me on my socials, I've got my Instagram, I've got my Twitter, and I also upload blogs. I'll catch you soon. Till then, Assalamualaikum.